These characteristics were used to qualify other statistical measurements used in later portions of the paper. It explains how they got approval for these different medical records and general permission. And then the polymorphism analysis was given through written consent as well from the patients. The second table gets more into the statistical research behind it. First, it starts off with a p-value relating the different genotypes to correlation between breast cancer. I demonstrated these in a figure. This figure shows that those who had breast cancer had a stronger likelihood of having either the TC or the CC genotype versus those who didn't have breast cancer kind of had a neutral between the different SNPs options. The chi-square test that was used defines statistical significance, and it showed a significant relationship between the genotypes of the SNP and those of breast cancer patients. This matters to us because it confirms the hypothesis of the paper. Another method that was used was an odds ratio, also showing statistical significance, and had a very strong confidence interval, which was a really strong, a very small range for the confidence interval for those who have the genotype of TC or CC. Both of these support the hypothesis that the SNP was correlated with breast cancer. Just to roll out the other factors, they also included table number 4, which listed p-values for other factors that were strongly represented in the population, using off of table number 1, which showed these characteristics. These different characteristics did not demonstrate a strong p-value or an odds ratio, further strengthening the argument that it was the polymorphism that has the highest correlation. There was also a chromatogram, which showed the sequences of DNA. This was cool because I got to learn what these were all about. They show a complementary sequence of the DNA, so the A for the thymosine and a G for the cytosine. In conclusion, things that we can discuss or further research that can be done is this SNP specifically susceptible to women who are Mexican, or is this to all races? Also, is this SNP supported by other papers talking about the same CYP 8A1? Or is it specifically this population, specifically this demographic, because it doesn't reach into other races? I'm excited to learn about how this affects me as a Caucasian woman, and also how genetics relate to breast cancer susceptibility as a whole.